Okay, so for this lecture, we are going to talk about gene exchange in bacteria. And so one form of gene exchange is going to be the movement of genes. These can be done by what we call transposons. And transposons are actually found um, in eukaryotes as well as prokaryotes. And they don't always result in a deleterious effect. And with respect to jumping genes in bacteria, they oftentimes are used to move around islands of pathogenic islands between plasmids, the bacteria genome, etc. So jumping genes are just what they say they are. They have the ability to move and they take things with them. So they can move within a genome, between plasmids, because remember, bacteria have plasmids, which you need to consider the plasmid as part of the genome of the bacterium. And they can also go between plasmids and genomes. They oftentimes are going to carry virulence genes, such as antibiotic resistant genes. They can, of course, insert randomly, so they will have some inactivation. But not all transposon jumping is going to result in deleterious effects. Most of the genomes, or a lot of the genome, has non-translated regions that the jumping genes can jump around into. They are going to be important for the evolution. They can facilitate translocation of genome sequences, shuffling of exons in our eukaryotes, etc., insertions. But for our prokaryotes, especially for our bacteria, it's mostly going to be pathogenicity islands. That the jumping genes are going to help to move around. So they'll flank the pathogenicity islands, and in this island it can have a capsule, it can have antibiotic resistance, etc. So an island of pathogenicity is just what it is. It has genes that have virulence factors, and they can move around using transposons. Now, another form of gene exchange is through plasmids. So plasmids are a way of the bacteria to transfer virulence factors from bacterium to bacterium, and there are some bacteria that can do this quite efficiently. So a plasmid is essentially a circular or linear DNA. And it's extra chromosome DNA. So bacteria have their own chromosomes circulating, and then they have these plasmids. And another thing about a plasmid is that they are self-replicating. So typically when a bacteria divides, it will give plasmids to each of its daughter cells, but the number of plasmid transferred to the daughter cells doesn't have to be equal. So some daughter cells might get more plasmids and some might get less plasmids. And these plasmids, again, are going to have virulence factors associated with them, and they're going to be able to transfer these virulence factors to their daughter cells, as well as other cells through exchange using what we're going to talk about is an F plasmid. But these plasmids have also been used for genetic engineering. So plasmids are commonly used to insert genes into yeast and bacteria. So if you want to make insulin, for example, you can use a plasmid to insert an insulin gene into yeast and they will make insulin. So these have been tools used in molecular biology as well. So now let's just talk about DNA transfer in bacteria. There are three main ways that bacteria can transfer DNA. There is through the process of transformation, and this is just active uptake. So if there is nucleic acids in the surrounding area, some bacterium are able just to take up that DNA, and if it confers a virulence factor, it's able to acquire that virulence factor. The second is through conjugation or mating, and this is the exchange, the active exchange of material between two bacterium. And then the third is transduction, which is a transfer of genetic information by bacteriophages, which are viruses that infect bacteria. So let's start with transformation. So transformation is basically you have a competent bacteria, and that competent bacteria can naturally take up exogenous DNA. So some of the examples of this are Haemophilus influenza, Streptococcus pneumonia, different Bacillus species, as well as Neisseria. And so basically you have a donor cell that dies, and the cell releases its own DNA, and then you have a competent cell that picks up the DNA that's been released by this donor cell. And again, depending on what the donor cell had, the competent cell will integrate virulence factor, antibiotic resistance, whatever it might have. Now here's an example of a competent cell and a non-competent cell. And so this is strep pneumonia. So strep pneumonia is competent. And I like this image because it kind of just shows what these bacteria have evolved to become. So you can see you have all these COM genes. That's for competent genes. And there's many of the competent genes, and we're not going to go through all these competent genes. But I just want to point out a few things. The strep pneumonia is able to release these little Pac-Mans. They're drawn like little Pac-Mans. And these Pac-Mans are really what are killing the non-competent cells. So strep pneumoniae actively kills bacteria around them, and they will kill non-strep bacterium because the strep bacterium have a competence gene, COM-M, which protects them from this little Pac-Man. So the little Pac-Man will go out and will destroy other non-competent cells, and these non-competent cells will release their genomic material, and then you're going to uptake the genomic material in the strep pneumonia. Now there is a very complicated pattern shown here. I just want to point this out that these are very complex interactions that these bacteria have evolved to do. But this is just to show that strep pneumonia actively can kill other bacteria, protect itself, and then pick up DNA from the surrounding that the bacteria died. Now the next form is going to be conjugation. Conjugation is going to be active passage of a plasmid from a positive cell, so a sex pilus F pilus positive cell, to a recipient. And then the recipient is going to be able to integrate that DNA into its DNA. So conjugation is going to transfer DNA from a cell that has the F plasmid and it goes from F plus into F minus, and then the F minus becomes F plus. So now it has the F plasmid and it can continue to transfer to other F minus bacteria. And so that's the process of conjugation. Now the development of MRSA, or methicillin and vancomycin, or methicillin resistant Staph aureus, or MVRSA, methicillin vancomycin resistant Staph aureus, is through the processes of both transformation and conjugation. So we have transformation, for example, we have our vancomycin resistant Enterococcus, and this has transferred through transformation to our staph. So the staph will actively uptake the DNA from your Enterococcus through transformation. Alternatively, if they have an F plasmid, they can transfer the information through conjugation, and then you acquire MRSA. And then MRSA, through transpositions, can acquire vancomycin resistant, or through a conjugation can continue to pass on both the vancomycin and methicillin resistant. And you can get the combination vancomycin methicillin resistant Staph aureus. And so this is a mechanism for acquiring or transferring resistance to a pathogen that is in the environment. So of course our Staph aureus is part of our normal flora, and acquiring these antibiotic resistant genes makes it more difficult to treat. And this is very important in a hospital setting because, again, we have a large percentage of people actually treating patients who have Staph aureus. And because we have patients that are immune compromised, the Staph aureus creates a big problem if they are antibiotic resistant. Now, the last form of transfer is through transduction, and this is going to be mediated by our bacteriophages. And a bacteriophage is, in essence, a virus, a bacteria virus. And so bacteriophages are a virus that will infect a cell, and then it'll cause cell lysis, and then the bacteria can infect an additional cell. Now, there are two types of bacteriophages. There's what's called a temperate phage, and there's a virulent phage. And there are two life cycles of phages. We have a lysogenic, and we have a lytic life cycle. And so our temperate phage can be both either lysogenic or lytic, and our virulent phage is always going to be lytic. Now let's go over these just a little bit. So basically for our lysogenic, some key things to point out is the bacteria survives, and it passes the phage to daughter cells. In the lytic, the bacteria dies. And so again, you can have a temperate phage that can either do lysogenic or lytic, or you can have a virulent phage that only does lytic. So basically in the lysogenic, you're going to have your bacteria or your bacteriophage inject its genome into the bacteria. And there's two options for our lysogenic. One is the phage bacteria becomes it circularizes and it becomes prophage, or your phage bacterium, just going to draw a circle with a few spaces, integrates into the host genome. So here's host. Either way, you're going to make new phage particles, and these particles are going to be released, and they can go off and infect. Now, alternatively, you're going to have these phage DNA, and they are going to be producing new phage particles. Times to the nth, and then this will burst and kill the cell, which is the bacteria. So typically for a phage, you have attachment, and this is typical of all viruses. You have some sort of penetration, typical of all viruses. You're going to have transcription, and transcription is going to lead to the lytic life cycle. For the lysogenic, you're going to have biosynthesis and incorporation. And then on the lysogenic side, the bacteria is going to be allowed to reproduce. On the lytic side, you're going to continue with biosynthesis of phage DNA. And then you're going to have maturation, and then eventually lysis of the cell and the death of the bacterium. So attachment's always 1, penetration is always 2. You have either transcription or incorporation, and then you're going to have biosynthesis, maturation, and lysis. And that's the end.